So in chapter 11, we're going to look at the meninges, the composition and function of cerebral spinal fluid. We're going to look at reflexes and what we call a reflex arc. We're going to look at major parts of the brain, what their functions are, the limbic system, peripheral nervous system, cranial nerves and their functions, sympathetic versus parasympathetic. So again, this is loaded with lots and lots of information. So be sure to soak this up really well. Take notes, read your chapter. I can't stress that enough. This information is somewhat, what's the word I'm looking for? More abstract concepts. Okay, and so we really got to kind of dig into these. So the brain is the largest and most complex part of the nervous system. It does have two cerebral hemispheres, the diencephalon, the brainstem, and the cerebellum. And the central nervous system is the brain and the spinal cord. The brain also houses about 100 billion multipolar neurons and countless branches of axons that actually communicate with each other. The brain stem connects the brain and the spinal cord together, and it does allow two-way communication, which is important. The spinal cord allows two-way communication between the central nervous system and the peripheral nervous system. So we have different membranes in there, and we call those meninges. And these are located between the bone and the soft tissue, and they enclose and protect the brain and the spinal cord. It's like a little covering. The meninges, or if it's singular, we say menix, M-E-N-I-X, have two layers, and actually three layers, and we call these the dura mater, the arachnoid mater, and the pia mater. Okay, and you are going to need to know these, all right? And so I want you to think the acronym DAP. Erase that here. Try the other side of my pen. It seems to work better. DAP, D-A-P, and that is going to be going from outside in. Dura mater, arachnoid mater, pia mater. Okay, you're going from outside in, D-A-P. So the dura mater is the outermost layer, and it's made of a tough, white, dense connective tissue. It has blood vessels and nerves. We have dural sinuses, which are channels formed where the dura mater splits into two layers. Remember, a sinus is an open cavity. Falx means sickle shape. So we have falx cerebelli and falx cerebelli. And these are sickle-shaped folded layers of meninges, all right? And then we have the epidural space, that is the space that lies between the dural sheath and the walls, okay? And so the epidural space is where you inject for an epidural, right? And so that's why knowing these layers, that's one of the reasons why knowing these layers is important. Dura mater, arachnoid mater, pia mater, outside in. So arachnoid sounds like a spider, right? Because spiders are arachnids. And so this is a thin web-like membrane. It has a web-like appearance to it. Has no blood vessels in it. It is located in between the dura mater and the pia mater. And it contains cerebral spinal fluid, which is important. The pia mater is thin meninges that's attached to the surface of the brain. It has lots of nerves and blood vessels which nourish the cells of the brain and the spinal cord. And then we can have inflammation of the meninges, and you probably already have heard of meningitis. That is the inflammation of the meninges. This typically occurs or affects the arachnoid mater and the pia mater, okay, but not the dura mater. So that's important as well. So this is showing you those different layers here. And so here on the left, they're taking a cutout portion here. Okay, and remember it's dura mater, arachnoid mater, and then pia mater. And so this is that blown up, all right? So here's the dura mater, all right? And then below that, you can see the arachnoid mater, and I don't want to draw a line because I'm afraid I'll cover it up. And then in between those two, you have some cerebral spinal fluid in blue, and then the pia mater. Okay, and so remember the falx cerebri and all, that is a folding of the meninges. All right, falx cerebri is a folding of the meninges and falx cerebelli. So you can kind of see, if you really look closely here, dura mater is very light blue, arachnoid mater is kind of pink. Then you have some cerebral spinal fluid there in blue, and then you have the pia mater. All right, and that's the meninges. Okay, and then it even shows you the gray and white matter of the cerebrum. So this is just a good example, kind of showing you the falx cerebelli and the falx cerebri, and how they're kind of, a sickle is that thing that you see the Grim Reaper has, that is actually used to cut things back in olden days. But this is just a good way to look at these. Like I said, I don't typically put a bunch of these images in here, but these can be really kind of hard to identify. So ventricles are cavities that are within the cerebral hemisphere and the brain stem, and they're kind of interconnected together. But we classify them or break them down into 4 sections. So we have 4 ventricles. This is important information. Be sure you know which is which when you are in lab, when you look at them, okay? Because those are often missed. They lie in the cerebral hemisphere and in the brain stem. They are continuous, or they're coming from the central canal of the spinal cord, and these are filled with cerebral spinal fluid. All right. So the 2 lateral ventricles are the largest, and we call those the first and second ventricle. The first ventricle is in the left hemisphere, which remember is going to be opposite on your patient, and the second is in the right hemisphere. The third ventricle is in the midline of the brain, beneath the corpus callosum. All right. And then the 4th ventricle is in the brain stem, so it's just anterior to the cerebellum. We have what we call interventricular foramina, or foramen. Okay, foramina are smaller openings, and foramen are larger openings, right? And so inter means within, ventricular, the ventricles, okay, or it means between, sorry, inter, between, intra, within. So between the ventricles you have some openings which allow communication to occur. The cerebral aqueduct connects those 3rd and 4th ventricles together. It is sometimes called the aqueduct of Sylvius, but I think you'll just see it as cerebral aqueduct. All right, so here you can see the ventricles. This is a really good diagram. I remember somebody stating that this looks like a phoenix or an eagle. And there was some way they used that to remember these, but I don't remember exactly how they did it. So maybe that'll come to my brain, or somebody will know, and y'all can email me and I'll share it with everybody. All right, so here we have the lateral ventricles here, okay? The lateral ventricles, all right? So we have one on the patient's right and one on the patient's left, okay? And then you can see here's the 3rd ventricle right there, all right? So 1st and 2nd ventricles, 3rd ventricle, and then this is the 4th ventricle down here. Okay, this one here, that's the 4th ventricle. You can see the aqueduct here between the ventricles. I don't think there's anything else too fascinating on these. All right, so the choroid plexus secretes cerebral spinal fluid. It's a mass of capillaries. They're specialized, and they project into the cavities of the ventricles and protects the brain from blows to the skull. Fluid is going to circulate through those ventricles and the central canal, enter that subarachnoid space below the arachnoid, and is then reabsorbed in the blood of the dural sinuses through what we call the arachnoid granulations. And you can actually see those granulations a couple slides back. I didn't point them out, but they are in some of the pictures if you look in your textbook. Cerebral spinal fluid is clear, or at least healthy cerebral spinal fluid is clear. It's about 140 milliliters, but humans secrete, the volume is normally 140 milliliters, at one time, but it's reabsorbed, so humans actually secrete a total of 500 milliliters of cerebrospinal fluid a day. Okay, but it's reabsorbed, so at any one time it's 140, but the total amount is 500, and that is important. So we have a high concentration of sodium and a lower concentration of glucose than typical fluids. The potassium is higher than any other extracellular fluid. And the purpose, of course, is to provide nutrition, and also has that shock absorbing protective nature to it. The fluid can even inform the autonomic center in the brain stem and the hypothalamus somewhat about the internal environment of the brain, which is really pretty cool. It can provide a pathway for waste to enter the bloodstream and protects the brain and the spinal cord. All right, so here we have the choroid plexus here. All right, and then here are those arachnoid granulations. You can kind of see them right here. The choroid plexus and the ventricle walls, remember, secrete that cerebrospinal fluid. Most of that fluid is going to circulate and enter that subarachnoid space where it's going to be reabsorbed into the blood. So there's the arachnoid mater, the arachnoid space, and so right below that. You can see it actually right here, the subarachnoid space. So the spinal cord is a column of nervous tissue. It is extending out from the brain and extends downward through the vertebral canal. The gray matter of the spinal cord is mostly composed or made of interneurons, and those are then divided into horns. All right. It begins at the foramen magnum, which is that large hole in the base of your skull, and stops or terminates at the first and second lumbar vertebrae in the space between those. All right, and then you can see here below we have what's called the cauda equina, which basically means horse's tail, because then you have nerve fibers that just kind of hang out low like that. So when you're looking at these diagrams, you kind of have to orient yourself to understand what you're looking at. This is a cross section of the spinal cord, and so you are looking down at the inside structure of the spinal cord here. Okay, and so we have some white matter here, okay, and then some gray matter. We have what we call the dorsal root ganglion, which is a gang of nerves, and then we have the anterior horn as well, and I have it blocked on my screen. Then we have posterior horns, lateral horns. Remember, the horn is just kind of like where it is sticking out, okay? And so you will generally see a lot of this kind of butterfly-shaped look, and that is representing the gray matter in the spinal cord. All right. Here's the ventral root down here. I didn't think I could see it, but it's there. So dorsal, think dorsal fin, like a shark has a dorsal fin, and ventral is going to be on the opposite side of that. So we have two functions for the spinal cord. It's the center for spinal reflexes, and it's also a conduit for the impulses that are going in and out of the brain. So it's a pathway for those. It connects the peripheral nervous system to the brain. Information in the form of nerve impulses reaching the spinal cord. They reach the spinal cord through those sensory neurons. They're transmitted in, and then they're transmitted up to the brains for the brain to decide what to do with that information. So we kind of know what an automatic reflex is, but we call that a reflex arc. Communication in the nervous system combines a series of action potentials along an axon, synaptic transmission between the neuron and the postsynaptic cell. It kind of puts those together. So reflexes are automatic, subconscious responses to stimuli within or outside the body. If you touch a hot stove, your reflex will be to pull it back. Okay, and so that occurs kind of without any conscious thought. So simple, sorry, I got tongue-tied there. well, before I go there, two or more neurons involved in the communication for the response to that stimuli are what we call a nerve pathway. A simple reflex or a simple nerve pathway starts with a sensory receptor and ends with an effector or a motor response. And so it can have as few as two neurons. All right. So this type of pathway is called a reflex. Reflex arcs, again, begin with a sensory receptor, and then they are processed in the central nervous system. We can have interneurons or afferent neurons that connect with motor neurons, and those fibers then pass from the central nervous system onto the effectors. And so this is kind of showing you that pathway. So you receive the information, it's going to go through a sensory or afferent neuron, send that signal to the central nervous system, which will then decipher that information, send a message back through a motor or efferent neuron, which will go to the effector, which is a muscle or gland. So this is a very simple pathway. So this is some really good definitions to understand these terms and functions. Effector, motor neuron, sensory neuron. So I'm not going to read this to you because you can read, but these are the basic components of all reflexes. So be sure you understand what these words mean and what the functions of these components are. All right, so here is a simple reflex arc. We have a sensory neuron in the central nervous system, and that's going to have at least one synapse, a motor neuron, an effector, and an interneuron. Okay, so this will be a spinal reflex, and the integration center is going to be actually in the spinal cord. All right, and so if we look here and we start, what color should we use? How about blue? And we start here, and you're sensing something here. Okay, that can be a touch receptor, it can be a cold receptor, a heat receptor, doesn't really matter. The point is it is sensing some information. Okay, that receptor is picking up the information, and then that information is going to go on an afferent or sensory neuron, okay, to the spinal cord. All right, and so this is the cell body of that sensory neuron, and it's going to go over here. Now, this little guy here is an interneuron, and it's just bridging the gap between the sensory neuron and the motor neuron. So the sensory neuron is the one that is carrying the information that has been sensed. And so you're going to do something with that, in this case in the spinal cord, because it's a simple reflex arc, and then send that information back out, that impulse back out that says, hey, pull your hand away from that hot stove. Okay, and so that is the effector. Here is the muscle. So we have the receptor, the sensory receptor. We have a sensory neuron. In this case, we have an interneuron. And then we have the motor neuron, which is going to send the information back to the effector, which is going to do the response that is needed, as in contract a muscle, which pulls your hand away from the hot stove. All right, so that's a very basic, simple reflex arc. So this is the patellar or knee jerk reflex, and this is an example of a simple reflex, and this is something your doctor does to test and see if your nervous system is working correctly. All right, and again, this is a simple one. It's only got two neurons. There's not even an interneuron here in this picture. So you hit that patellar ligament right here. When he takes that little hammer, hits that patellar ligament, and a sensory neuron is going to pick up that pain sensation and communicate that all the way up here to the spinal cord, and then it's going to send a message back through the motor neuron. Okay, and that is going to cause your quadricep muscles to be pulled, and that triggers stretch receptors, which send impulses on to another unipolar sensory neuron, and that causes that knee jerk response to occur. Okay, so if you'll notice, these are color coded. All right. This is a simple monosynaptic, which means one synapse reflex. I'm sorry if you hear my dog snoring. If you have not taken me for Anatomy 1, you might see lots of little critters here in the videos. Sometimes one will jump up on the bed, and sometimes you'll hear the bigger one snoring. So I apologize if you can hear her in the background. Anyway, so this can also help maintain upright posture without thinking about it. If somebody comes up behind you, have you ever had somebody come and take your knee out when you're standing there, and then your other leg automatically tightens? We'll look at that kind of in a minute, but it's kind of like that. That's a crossed reflex. But so anyway, you feel this. The sense here is going to go up to the spinal cord, be dealt with and sent back, and tell your muscles to tighten up. And you're not thinking about it. So this is a withdrawal reflex, and this is the one where I'm talking about we touch the hot stove and it causes a withdrawal. So the person encounters something painful that involves a sensory neuron. You also have a little interneuron here. You can see this interneuron here. I think that's in purple. And so you step on a tack in this case. All right, and so here the impulse is going in. That's going to go through sensory, all right, go to the interneuron, and then be relayed back through the motor neuron, all right. And this is an instinctive type of thing to prevent tissue damage. So this is a crossed extensor reflex, and you can see it's a little bit more complex, right? I've got several things going on there. And this is what happens when somebody comes up behind you and takes out your knee. What tells your other leg to support your weight, all right? And so that's a crossed extensor reflex, and that's the crossing of sensory impulses within the reflex center to produce the opposite effect. So when the flexor muscles on the affected side, we would call that the ipsilateral side, contract. The flexor muscles of the limb on the other side, which is contralateral, are inhibited, so they relax. So you got to have a tightening of one and a relaxing of the other. So the extensor on that contralateral side contract and help support the body weight. That's a crossed extensor reflex. We've got interneuron pathways in the spinal cord. All of these sensory impulses that come in on one side of the cord go to the other side to produce the opposite effect, so that can work together. So it's really pretty cool, because if you tighten one and don't release the other, or if you pick your left leg up and don't tell your right leg to tense up to hold your weight, then you would fall. So we have descending tracts or pathways and ascending tracts or pathways, all right? And so this is a pathway that carries information. We have corticospinal tracts that carry instructions that control voluntary movements. And then the reticulospinal tracts control muscle tone and activity of sweat glands. And then we have what we call the rubrospinal tract, and that coordinates muscle actions. So knowing where these are and what they do really helps with diagnosing problems. All right, so let's look a little more closely at the brain now. The brain is really fascinating. The brain has areas that are associated with particular functions, sensations, smells, perceptions. It issues commands to the skeletal muscles, and it performs higher mental functions like memory, reasoning, and coordination of muscle movements. It also regulates visceral activities and oversees functions of the entire body. It gives us all the characteristics that actually make us who we are and give us our personality. All right, so there's a little bit more thorough list there. We have the different functions of the brain, but let's look at the major parts of the brain. And that other picture actually was probably a pretty good one to look at for that. I would actually recommend printing or drawing of the brain and color coding those. So you've got your frontal lobe, your parietal lobe here on the side, temporal lobe, and the occipital lobe. Well, the one that is the hardest is called the insula. Insula means island. And so that is this green portion inside here. So that was one that is often missed, all right? So be sure you remember that one, okay? Then we also have the diencephalon, which is in purple here. I think that's purple. Then we have the cerebellum. Cerebellum means little brain, so it almost looks like a little mini brain underneath the cerebrum. And then we have the brain stem, which is very important, and that's this point here. And it is formed by the midbrain, the pons, and the medulla oblongata. And we're going to look at those. So here are those four major portions. You can see the cerebrum and the cerebellum, but more importantly, I wanted you to see the locations of the midbrain, which is part of the brain stem, the pons, and then the medulla oblongata, okay? And then here is the diencephalon. All right, so our brain has two hemispheres. We often call it the right and left brain. The space in the middle, or the fissure in the middle, let's find a different color here. Right here, you can see that here is the longitudinal fissure. It's a longitudinal cut. And then we're going to look in a moment at the gyri and the sulci. And then, of course, we have the somewhat C-shaped corpus callosum, which joins the two hemispheres together. It helps coordinate, the cerebrum helps coordinate voluntary muscle movements, as well as many other things. All right, so this is a sagittal section, or you can think side view for sagittal, of the brain and spinal cord. And it's the uncut medial surface of the right cerebral hemisphere. And again, you can see the major portions of the brain here, cerebrum, the diencephalon, cerebellum, and brain stem. So these are just some good pictures to look at for that. So when looking at these in brain development, and we're talking about embryonic brain development as you are forming the brain in the embryonic stage while it's still in the mother's womb, why would that matter? Why is that important? Well, that is important because you can have defects that occur when this process doesn't occur correctly. So understanding the process helps us to understand the defect and therefore treat the patient, okay? So the brain structure, or the basic structure of the brain, I'm getting tongue-tied, reflects the way that it formed during embryonic development, okay? So it begins as a neural tube that gives rise or changes into the central nervous system later on. The portion that contains the brain has three major cavities, and that's the forebrain, which is called the prosencephalon, the midbrain, which is called the mes means middle, pro means before, so fore and mid, and then the hindbrain, which is also called the rhombencephalon, all right? So later on, then, the brain divides more. It divides into anterior and posterior portions, and the hindbrain divides into two parts, which gives us 5 total cavities, all right? And so the 5 secondary cavities are the telecephalon, which is forebrain, from the forebrain, and then the diencephalon. So the forebrain, or prosencephalon, diverges into the telencephalon and the diencephalon. All right, and so you can see it just kind of starts to differentiate, is a good way to look at that. Then we have the mesencephalon, which means middle, metaencephalon, which is comes from the hindbrain, and then the hindbrain also splits into the myelencephalon. Okay, and so the top and the bottom split a little bit more. So in order to show you why this is important, or the effect of these not separating out correctly, we're going to look at a few of the birth disorders that can happen, or developmental defects that can happen. So a fetus or a newborn can have what we call anencephaly. And this is where the face and lower brain structures are there, but they lack most of the higher brain structures. And this is a neural tube defect, okay? A newborn with this anomaly only survives a few days. It occurs at about the 28th day of prenatal development. And how do we know that? Well, we know that because that's when some of these splits occur. So a sheet of tissue that normally folds to form that neural tube, which is going to later develop into the central nervous system, doesn't fold. It stays open at the top. Okay, and so that causes anencephaly. And we're going to look at pictures of these, and they're kind of hard to look at, but I want you to understand why that neural tube and the way it changes, why is that even relevant? Okay, because how else are you going to learn it if you don't understand it's relevant? And so then we have spina bifida, which you're probably a little more familiar with. And that is when an opening is further down the neural tube than it should be. And so if it's open further down, it can actually cause a paralysis because it doesn't close up. So it can cause a paralysis from that point downward. In some cases, surgery can be done before, so while still in the womb, or shortly after birth, and correct some of that. So that is really, really important. This is why prenatal care is so important, because if you don't catch it, you can't fix it. Folic acid, the reason they pass that out so much now is this can help prevent this from occurring. This can lower the risk of it occurring. So likely there is a reason, if there's a lack of folic acid, that this process doesn't occur. And the last neural tube defect here is lysencephaly, and that means smooth brain, and this is where there's actually a smooth cerebral cortex. There are no gyri or sulci, and the gyri or sulci are the infoldings of the brain, okay, where they're sunk in and where they're kind of convoluted around. And so they don't have any of those. And so there's a protein in early prenatal development that can actually prevent certain neurons from migrating in the brain. And those normally will stop actually, that's going to cause the convolutions to not occur. And so the child then is going to be profoundly disabled, have seizures, and lots of other neurological problems. So understanding when these problems occur helps us to be able to prevent and treat them. This is why we know to give that folic acid. This is why early prenatal care is so important. Taking those vitamins is so important, because if you're lacking that protein and you're eating right, and you take a vitamin that has what you need in it, that folic acid, and you can stop this from occurring, of course we would do that. So here on the left you can see these two pictures are of a child that is born with anencephaly. On the top right we have spina bifida, and then on the bottom is the lysencephaly, or the smooth brain, and you can see there are no convolutions, there's no infoldings. And the problem with that is it decreases surface area. The more surface area we have, the more material we have.